Oh, yeah. So what I do is I'll just complete what we were talking about yesterday, and from Tuesday, Wednesday, and Thursday, we'll go through the next 2 units, which would be um, sensation and perception and the motor systems. So that would take a little more time. So I think those 3 classes should be sufficient for those, and then I think 2 more classes would be left, and then we can finish off memory system, synaptic plasticity, cognition and disorders, and brain diseases. So I guess there are totally 10 classes. You all can hear me, right? Can you hear me?
Yes.
Yes, sir. Yes.
Okay. Yeah, so that's that. And so today, as I said, it'll be pretty short. I have some other work. And so first what we'll do is I'll show you guys something that's I think is very interesting. Okay, so this is what you call this website is maintained by Allen Brain Institute. It's in Washington State, in Seattle, in US. And so this institute was started by Paul Allen, who is one of the founders for Microsoft. And he put in a lot of his own money into neuroscience research. So what they have done is they basically, started this, or like, I think 2000, middle 2000, like before 2010, 8 or 9 or something like that. So it's like 10, 12 years old. And what they tried to do was, so they thought that, neuroscience should be developed really fast, and it's really important to understand how brains function. And the way things were going, that all the individual scientists, just like us, like people are working all over the world towards kind of their own goals, and the things were very slow. And the idea was that if somebody comes with a lot of wealth and put the money into it and get the best scientists together, hopefully you can, because most of the science that's done is publicly funded, which means that it's funded by taxpayers' money. So you kind of you have to follow what's important today, and you have to study those things. But how do you study just basic neuroscience and kind of try to accelerate the state of research by say 20 or 30 years? And they actually have successfully done that in 10 years. And also what they have done is, the problem with individual groups working all over the world is that they have different principles, they have different priorities, they have different biases. So how do you develop it very so that you develop it in a way that everybody else in the field can trust it? So what they've done is they have done huge number of experiments which are quality controlled, just like industry, just just like a product is developed. And not a single person takes credit for it, but the whole organization takes credit for it. And since they're funding their own research, they can decide basically what they want to do, and they decide what is the most important thing that needs to be done, and they just do that. And so when I was talking about neuroanatomy, so the gross anatomy is kind of fine. We all agree with it. But when you look at the finer aspects of it, since it's a huge tissue with billions of neurons, we need to be very consistent where the cell bodies are, where the axons go, where the exact circuits are, what are the molecules that are expressed in it. And so again, like the reagents between different lab can be different. They can have contamination. So often one group will argue with the other and they won't agree with each other. So they wanted to, so I just giving an example, they have done many experiments like this. So I'll just give you one example from it. So this is called Mouse Brain Atlas. And if you remember the last slide of the last class, and what was it I showed you, in situ hybridization. So you're basically looking at gene expression or levels of mRNA of different genes across the brain. and then you can map it to the brain structure. So it's something like, you collect data, say like you have a Google map, your not Google map, like you have a map of India. Then you collect data, okay, like say there are 500, there are, say like you were going for a surname, like XYZ, and then you want to map out how many people of those surnames are living in which city and which village and exact geographical location across the country. So you collect the data and you basically map it out to the country's map, and then you have this whole map of the country saying that, okay, with this surnames, this is the distribution, graphical distribution. So you want to look at what they have done is they have gone through the whole genome, which is like all the genes in the genome, so which is basically like around 30,000 genes. And they have done in situ hybridization for all 30,000 genes uh, across the brain. And what they've done is first they developed a macroscopy kind of, what do you call that, like a workflow where everything is automated. So they don't, that's it. So it's not, they just quality control it just like industry. So they process the brain the same, every brain the same way. They repeated the experiment. They, even though like the microscopy, the imaging part is automated. So the brain gets processed from the point that the brain left the mouse's head and the mouse died. From that to the images, everything is automated. So it's cutting by itself, it's imaging by itself, and it's all done by robots, right? And then eventually, when the images come by, they curate. So that there are scientists, neuroscientists who have done PhDs and or postdocs. So they would now curate the images, make sure they're looking at the machine, what machines have put out, and they will decide, okay, this is okay to be put in the database and this is not. So, and what has happened is last 4 or 5 years, all the scientists are using this data and they're seeing that they're extremely consistent. And in most cases, they're kind of downplaying the data. So they are kind of very conservative about the conclusions they want you to make from it. So they're almost all, like every single time correct. Maybe they're giving a little less information, but they never gives out. So it's a very, what I'm saying is like, they have changed the face of cellular molecular neuroscience forever and accelerated by 10 or 20 years. So I'll just give an example. So say, one of the genes we're talking about is GLI2, which is basically a transporter and which kind of loads the synaptic vesicles with the glutamate, which is the neurotransmitter. So we'll just can just search for GLI2 and search for it. And so they will show you 2 experiments they had done. One is a sagittal and one is a coronal. And if you remember, how you can cut the brain 2 ways. One is coronally, that is across, and then sagittally. I'll show you how it looks like. So you click on the experiments and you go here. Oh, you can see that. Yeah. I have to show you here. Anyways. So this is in situ hybridization. So darker is where the genes are expressed, and this is the 3-dimensional kind of model of the brain. So they have imaged every single section, and they have kind of using algorithms, they have reconstructed the whole brain, and they have done this for every single gene in both ways, coronally and sagittally. Some experiments they have repeated many times, and this shows the expression of the genes that are different levels of genes at different areas. This is the when you do in situ hybridization, you basically use 2 primers, which is basically the sequences with this. They kind of amplify this so that you can eventually visualize that. So this is the part of the gene that they selected to amplify, to probe against, or so that this is very specific to BGLU2, and they are not kind of detecting anything else. So you can also repeat the experiment using their the information they have given, and you can confirm by yourself what they have done is right or wrong. They also give you the data information about the mouse. They use the species, the age, and gender, and everything. So it's done very professionally. Yeah, and then what this is, as I said, so this is cross section. So you basically going from this side to this. So this is the one. This is the coronal sections, and you're going from rostral to caudal, from the front to the back. So this is olfactory bulb. This is the cortex, and the midbrain is inside it. So this is and then the cerebellum and the pons or the brainstem. So you're going from forward to backward. And also what you can, so you olfactory bulb, so of course not every section is perfect. So it looks sometimes absurd, but it's pretty much really nice. But yeah, so you can see that this brain region, whatever this is, you can see expression here. And so you can go more. You can see, the most of the cortex, like here in hippocampus. So this is the thalamus, hypothalamus. Same thing. You basically see the expression. Need to share this one. Yeah. So if you see here, you're now seeing the different brain regions I was showing you before. Okay, go a little forward. Yeah. Yeah. So there's a section, and then they have actually mapped it out to the brain map they generated. So I'll show you that. So this is the key for the brain map. You can zoom in more. And then you can do this. So, and this tells you the exact brain region. So they go to subnuclei. So this is called thalamus. So thalamus has some 50 or 60, approximately brain nuclei. And then you can see, so the name shows up here, mediodorsal nucleus of thalamus lateral part. And each of this actually have a paracentral nucleus. So there's a cortex, there's a different layers with the development of which you're talking about, where, when the mice are developing or as embryos, the neurons basically migrate from this ventricle and they go here and they kind of go up like this and settle along different layers. So these are full of neurons. So this blue color is it's just showing all the neurons. And so there's the hippocampus and cortical development. I'll get into a little bit, in the next slides, I show you. And so this is that. So this is full of neurons, but you see only a few number of neurons are expressing VGLUT2 because there are different kind of transporters. That doesn't mean that the other neurons don't express glutamate. But this is only one of the transporters. And other advantage of it, which we'll get into a little bit at the end of the course, is that how do you use this information for doing experiments? So what we do is we make different transgenic animals that are as I kind of gone through a bit before that. How is the cell right here deciding to express VGLUT2 or not? It's decided by this thing called promoter. So if the promoter is just before the gene, and the promoter is active only in this nuclei and not in the next nuclei just next to it, not in the neuronal nuclei just next to it. Hence, it's expressing VGLUT2. So what it is that we can take advantage of this fact. Now we can just move the VGLUT2 and the gene, the coding region, we give the promoter intact, and instead of VGLUT2, we can express a fluorescent protein like GFP, and all the VGLUT2 neurons will light up. Then you can ask questions. So instead of GFP, if you have like a calcium sensor I was talking about yesterday, you can record the activity of the neuron. So you will be able to record only from the VGLUT2 of the neurons of the in the cell, and you won't, and since you are using a microscope, like you won't be able to see anything else because only those cells are expressing the fluorescent protein. What it does is it gives you a kind of access to cell cell type specificity. So now, so for example, there is a molecule is expressed only in this nuclei and nowhere else. Now, if you kind of put a lens here and you imaging from it what the neurons are doing, then you won't see any other neurons. So everything is silent, which is this advantage, a great advantage this kind of techniques provide, which electrophysiology, which you basically kind of blind to it, you're basically poking any neuron, cannot do. So now we can ask, okay, we can now then divide by the molecular identity. Say there are 100 neurons here. We can actually divide into 5 parts. And then we can ask what are these 20 neurons are doing in respect to other 80 neurons or so. So you can go more, you can even divide this, anatomical nuclei into many parts with their function, where they project to, who they're giving information from, and who they're giving information to, and etc. etc. So basically, like what I'm saying is, you can we take regularly advantage of this data and design experiments similarly. Yeah, and so that's that. I just wanted to show you that, and then we go to the slides for a bit. Yeah, so I have only a few slides, 4 or 5 slides. Can you see the slide now?
Yes, sir.
Can you see the yeah?
Yes.
Yeah. So no, this should be the 5th. This is the lecture 5th. Anyways, so yeah. So what I'll go in very short, as I said, like today, we'll keep it very short. So how are human brains special, right? So, we talked about most of the studies that I'm doing. I mean, there are a lot of there are a few human neuroscientists who are using human subjects and primates at Center for Neuroscience. But I'm a mouse scientist. I'm a rodent neuroscientist, and the advantage of being that you can do a lot of things with rodents which you can't do with humans, obviously, but if not even primates, because they are much bigger animals. You can't just they're very valuable, they have emotion, so you can't just and they can express the emotions, right? So you have to be very careful when you're doing experiments with monkeys, and also they can kind of display very complicated behavior just like us. So and also you can ask them questions that you can't ask a mouse. They're much more agile, they're more human-like, and they will do things that mice or rat can't do. But then there are advantages and disadvantages. But anyways, so what are the specialities of a human brain? So this is the size difference. This is how relative size of it. But if you kind of make them equal, these are the different structures. So the most striking feature is you can see the cortex. There's the folded structure at the top. This completely covered the rest of the brain in the humans, which it doesn't in in rats. So the cerebellum and the brainstem is not covered by the cortex at all. However, in humans, the cortex is just huge, and then they have these poles which are called gyri and the sulci, which we think scientists believe or not believe, but which we kind of know, but we don't know. I don't think we know exactly why, but at least we understand that during evolution, because we have so much computing power in the brain, we need a way bigger cortex and way more neurons, and which have transformed themselves. So since the neurons are there's so many neurons, so it is folded down to increase the surface area, so that the brain can house so many neurons, which is way more than most other animals, or most other even mammals. And And the other thing is that, so the relative size now have decreased. So most of the brainstem and cerebellum are kind of, so cerebellum is also a little more differentiated in humans, but otherwise the basic cells and structures are kind of conserved, except for this relative size and relative differentiation. So, the cortex is like hugely different, to the point that, a lot of scientists who study with humans and primates, they don't even believe that other animals actually have a cortex. They think this is just, nothing. So, but yeah, so that's where the differences are. And then the olfactory bulb in animals, so we have a much less developed olfactory bulb. And as you can imagine, like a lot of, mice, rats, and cats and dogs, and they use the nose to their olfactory system to guide a lot of behaviors where they find food, or they find kind of expect where the preys are coming from, or where the dangers lie. So they use their olfactory system a lot, and so their olfactory system are much more developed than ours. So it's relatively less developed. Otherwise, like pretty much structurally, it's similar. But the fact that it has so much of cortical, so many of cortical neurons, is more than any other animal. So the assumption is that most of intelligence and conscious perception of things actually come from, or agility, motor control come from this cortical neurons. And so we're talking about development. One second. Sorry. Yeah. So as you talk about cortex and cortical development, and in the adult brain pictures, I showed you the different. There's a here, but there are different layers of the cortex, and how do you start? So the if you look at neurodevelopment and you look at advances of neurodevelopment, so one of the most fascinating story that comes from the cortical development. And why you study cortical development, as I said, that in humans, that's the most evolved part. So that's what differentiates us from structurally and anatomically from most other animals. So if you want to study development, how the brain comes to like kind of is formed and is assembled, then probably studying the cortex is is the smartest thing to do. So I just want to point out, you guys can go and just Google him and kind of watch his videos and what he has done. And I think he's a fascinating character. And what he had done was he was one of the first people like who kind of, so Kahal, as I went to the first class, he started drawing out cortical development and he was very accurate. But what Bhaskar Rakesh started doing was, so he was assistant professor at Yale, and he's kind of active for the last 40 years or so. So what he had started doing is drawing out. So he kind of from aborted fetuses, he started doing kind of looking into how cortex is developed in humans, and he wanted to make a framework that so that people kind of everybody agrees to. It's kind of something like adult brain atlas. And so there a conference they call the Boulder Committee, and he proposed like we kind of follow a similar narrative, like similar naming of how cortex is developed. Yeah, and then so in the Boulder Committee they agreed of a common framework, and that's what they proposed, and he revised it later in 2008. So this was in '69. So he basically what he did was he looked into the different cells. So these are the different layers. So this is called cortical plate, the intermediate layer, and the ventricular layer. So the neurons, this is the base of the cortex. So the neurons are coming in from ventricle, and they're slowly going up on the radial glia and ending up where they're supposed to be. And then they have the dendrites at the top, and their axons are going all over the brain. So the axons will go from here, so if you can imagine, so this is the whole cortex, like a kind of a semicircular kind of pattern, and then these axons will go around across the cortex and across the whole brain, and kind of the information the neurons are getting, they'll process them and kind of provide them, kind of give the feedback what to do about it. And then, so here there were some edits, and they found different things that different. So they added more cell types into it. So there are, radial glia cells. There are different kind of neurons, different kind of cells, neurons that are, populating different layers. So, and then I would just like to show you guys a little bit, how what is the current state of affairs is. So, so what they do is like this is how the experiments look like, right? So this is embryonic 15.5, that is gestation 15 days. So as I said before, 21 days is what it takes for a mouse to be in the womb before they're born. And so you can depending on gene expression, you can look at different types of cells during development. So they have this is the radial glial cells, which are the neurons used to climb up. And so there is the ventricular zone and the cortical plate I was talking about. So I mean, all you need to understand is that you have the really kind of giving the birth for the neurons to go up. And so what they found was quite interesting. Like so they knew that the neurons are basically the neurons that climb up, and how they fire while they are going up. It determines the position, how they're forming connections with each other. So there's a lot of work being done is how the neurons are actually setting up what are the molecular mechanisms. If you take out a molecule, they can't go up. Then the neurons actually like kind of coordinate with each other. They fire, and then kind of they form the connection. So once they have to migrate, they have to settle down at a position. Then they have to make connections, and this is very important because in every animal, most minds are almost similar. So in every animal, most of these connections will be very different. They might change by experience, but the basic structure, underlying structure and function should be very similar. And this is done, if you can, I mean, it's incredible that in every animals in humans, every single species of the earth, in animals, every single animal within a species on earth have very similar patterns of development and molecular mechanisms, and where the neurons are residing, whom they're talking to, whom they're making connections. So, and this is all happens by itself, and so it's a fascinating field. And again, like quite scientists are studying that thing. So one of the kind of more recent findings they had was they looked at so they knew that the neurons are firing, and the neurons while they kind of migrate and they develop and they form connections. They even found that so if you see this, that the glial cells or the radial glial cells also fire. So so now imagine like how, so these are not just kind of railway tracks, like dead railway tracks, but these glial cells are also kind of deciding for the neurons where to go or where not. So it's an incredibly complicated environment, and incredible number of the point is that there are so many variables right now, incredible number of variables that the neurons depend on how to form connections, where to form connections. And in most cases, as you know, that in most cases we're all normal, and we don't have developmental defects. And so for most cases, it's a very precise method by using which these neurons can wire together. And and then another topic is kind of synaptic plasticity. So what is synaptic plasticity? Yeah, so what is synaptic plasticity is basically the synapses are, I talked about before, they form the connection once, but they're very dynamic in nature. So the video I was showing you, the dendrites, they come and go, and they decide, learning happens at these dendrites. And so there are various theories that decide, how there are various theories that decide how neurons are connected and how they're plastic. So after how much activity, the neurons will actually finally kind of remember something, or will remember something through the synapses that are kind of being changed in terms of molecular composition and anatomical structure. So one of the theories that it's called a Hebbian theory. So it says that if the sorry, this is neutrons, but if the neurons fire together, they are wired together, which means that if you're saying that if you're seeing that the two neurons in the brain, they are kind of firing very close to temporally very close to each other, most likely they are also connected. So which means that which also goes that that's the theory of the theory of synaptic plasticity says that if it repeatedly goes on firing, their connection will basically get stronger. And and more recently, there's kind of concepts like LTP and LTD, which are called long-term potentiation and long-term depression. You don't have to know much about it, but what it basically is, what these terms basically mean are that how does learning happen? So at the level of the synapse, so how do we, if we learn a new thing, how is it happening? And if you can imagine, if you're learning and say you are learning how to walk, you can't just try once and learn. You have to go and trying again and again. So the idea is that if the neurons that are required for your learning to walk, they need to fire again and again and again. And once the synaptic kind of the plasticity is solidified or is kind of the synapses have molecularly cellularly changed from one pattern to the other permanently, then you have learned how to walk, and you don't just go away just like that. So how does that happen? And at the synaptic level, which is called the long-term potentiation. So if the neurons are the postsynaptic is providing action potential again and again and again, eventually the potential difference between those two neurons will alter, and will just stay there like that, and which is called long-term potential. And then there is long-term depression, which is basically the opposite of long-term potentiation. Is when you need to kind of revert back, reverse to the original from a changed kind of strengthened synapse to a weakened synapse, and you have something called long-term depression. And a lot of scientists are studying the molecular mechanisms of this using a number of techniques. So that's that. And then, as I said, so from next week, we'll go to sensory and motor systems, and we'll try to finish in the 3 lectures next week. Okay. Yeah, so that's that. And questions? Yeah, anybody? So anybody has anyone? No? Yeah, Vinod.
No, I don't. So they don't have a.
Okay, so we can just close it there. All right, thank you.
Jean, you want to discuss with us regarding exam, right? Hello, Jean. Jean, we couldn't able to hear you.
Hello. Hello.
Yeah,
Yeah, now
yeah.
it's audible, no? Yeah. Dear students, so actually we are planning for the practical exam. So I think Saturday everyone is free, right? Hello.
Yes,
Sir, are you talking about the first two experiments?
Yeah, the first two experiments. So combinedly you will have an exam. Okay, we are planning for exam combinedly. So 15 questions from the first experiment and another 15 questions from the second experiment. So I heard like that your immunology practical exam has been postponed. So I think we can make use of this Saturday. Otherwise, no, you'll be just like packed for the next 2 weeks. So it's very difficult to find time in between. So it's better to give the exam by this Saturday. So what are you thinking? So we can give the exam, no?
yes, sir. That's fine.
So Saturday people are free.
Sorry, I didn't catch
So
you.
only so Saturday people are free, no? Students are free, no? Can
Yeah,
I
yeah,
fix
yeah.
the time by 10 to 11 to 12 or sometimes we have 11 to 12.
Sir, will it be a Microsoft Forms thing where you have MCQs or we have to like write and submit or?
So I don't know the exact structure of the thing. Most probably it will be an MCQ. MCQ. That will be MCQ. It will be in
So
Google.
it'll be completely online.
Yeah, it will be completely online. So that's the problem. If you are not convenient with this timing, or just later if you are joining, then again we have to set the questions and all. So can we fix the time by 11 to 12? So if you have some problem with the timing, just we can reschedule a little bit of it, something, just the problem. So it's coordinate from the neurobiology department also. So getting just like a common timing is very difficult. That's why.
Sir, so please put this, what do you say? Put the timing. What do you say? The timing you want to give. Please